The overall goal of this videomicroscopy technique is to examine the functional properties of isolated microvessels in response to pharmacological and physiological stimuli, providing insight into the pathophysiology and molecular mechanisms that contribute to vascular dysfunction in humans.
This method is useful for understanding the molecular mechanisms that contribute to dysfunction of the local microvasculature within fat, which has been linked to systemic diseases. The main advantages of this technique are that blood vessels remain functional after removal from the human body for a period of time and are easily examined for their physiologic properties.
Clinical relevance of this experimental approach is that it allows us to identify pathways that are differentially altered in disease conditions and to potentially discover novel therapeutic targets.
Under a tissue dissecting microscope, use microscissors and microforceps to carefully remove the surrounding fat and connective tissue from the small arterioles within the adipose sample.
It is essential to distinguish the arterioles from the venules. Arterioles are typically smaller in diameter, demonstrate a greater tone, and respond more robustly than do venules.
When the arteriole has been isolated, use nylon or silk sutures to tie off any branches. Next, use a 10 ml syringe to slowly fill the tubing with fresh Krebs solution. Then, attach rubber tubes to the pressure reservoirs and glass capillary pipettes within the chamber. Next, move the